Hi, class. So today we're going to be continuing on with the catabolism of carbohydrates and discuss how these carbons are typically used to be brought into the TCA cycle, how electrons are harvested, and how the reaction chemistry works with the TCA cycle. Now, this is one of the most central metabolic pathways in all of metabolism, and understanding it is really critical for understanding how we connect all the metabolic pathways in the body with biosynthetic reactions, and it's the crossroads for metabolism. So the overall roadmap for today is we're going to talk about the bridge reactions between glycolysis and anaerobic and aerobic metabolism. We're going to talk about the TCA cycle reactions themselves, and we're going to talk about anaplerosis. So as an overall concept map, just as a reminder, we're talking about reactions that primarily are taking carbohydrates, sugars in one way or another, and bringing them into the TCA cycle, okay? Which isn't to say that this is the only source of carbon that can enter the TCA cycle. Carbon can also enter from proteins, and carbon can also enter from fats. And we'll be learning more about those mechanisms in a bit, but this is following on from our previous lectures on carbohydrate metabolism. And as a connection here, this should be familiar to most of you, is that glucose has come in. We ultimately have pyruvate. That was the endpoint of glycolysis. Pyruvate can ultimately be turned into lactates created via anaerobic reactions. It can also be transaminated to become an alanine, or this fate is it can be converted into acetyl-CoA, and those carbons enter the TCA cycle. Now, this happens in a couple of steps. So first step are there are these bridge reactions that bring the carbon into the mitochondrion and prepare it for further oxidation. And then there are these oxidation events that harvest electrons off those organic scaffolds and ultimately pass them along to the TCA cycle. And the overall reaction here for this entire process is here. We have an acetyl-CoA, 3-NAD, an FAD, an ADP, an inorganic phosphate, and two water molecules, ultimately becoming 2CO2, 3-NADH, FADH2, ATP, proton, and a CoA. Now, these obviously can get further converted into, they can donate their these hydrides along to electron transport chain that can be used to drive ATP synthesis via oxidative phosphorylation. And that's these reactions that are shown right here. So again, acetyl-CoA comes in, that has been ultimately used to harvest these high energy electrons, and they're passed along to oxygen to make water and pump protons against their gradient, and then the leaking of that proton back into the mitochondrial matrix, or in this case, the intracellularly in the case of prokaryotes, is used to drive ATP synthesis. Now, we will learn these things primarily in the context of eukaryotic cells and think about them in the context of human metabolism, but these are reactions that are shared amongst almost all living organisms. So prokaryotes have these reactions too, and the only difference is that prokaryotes don't have this happening in their mitochondrial matrix, but instead have this in their happening along their cell membranes there in the periplasmic space. Okay, so the bridge reactions. First and most important one is the pyruvate dehydrogenase complex. So this converts pyruvate into acetyl-CoA, and the process makes an NADH and releases a CO2 and a proton. Now, this is a really complicated reaction, and we're going to discuss a little bit why. There's so much machinery involved with this, but there are three major subunits: pyruvate dehydrogenase complex here, this dihydrolipidyl transacetylase, and this dihydrolipidyl dehydrogenase. And each of those has their own cofactors: thymine pyrophosphate, lipolamide, and FAD. And we'll talk about how each of these steps work in a moment. This is the overall schematic as shown in our textbook. which is showing pyruvate entering, losing the CO2 in the first step, and interacting with this thymine pyrophosphate to make this acylated version of the molecule. That's ultimately transferred on to this lipolamide to make this acyl lipoyl lysine here in the membrane. That's ultimately transferred along, and a CoA moiety is then used and attached onto this acyl group, and the acyl CoA is what's ultimately used in the mitochondrion. So this entire process leaves a reduced lipolamide here, and so to get this usable again, the enzyme uses a coupled reaction between FAD and NADH. Ultimately, this is coming out as NADH to oxidize this guy back into its original reactive form with the disulfide bridge. And that's what ultimately these three units are formed. Doing the decarboxylation reaction, transferring the acyl unit onto a CoA, and then three is regenerating the lipolamide for further chemistry and harvesting this NADH. Why is there so much machinery involved in this? The reason for that is that in order to do this chemistry, we have to do something that we've talked about as not being possible before, which is that, or not spontaneous before, which is that we have to break this bond here, which is alpha to carbonyl, right? So normally we talk about bonds beta to carbonyl being labile, but in this particular instance, we're trying to break this bond and release this CO2. In order to do that, we go through this chemistry with this thymine pyrophosphate that creates a circumstance where we have this double bond placed beta to a carbonyl, and that enables resonance to occur. And let me show you how this works. So you have a nucleophilic attack. First, the base abstracts a proton from the thymine pyrophosphate, is activating this as a this molecule as a reactant. That leaves this carbanion, which can serve as a nucleophile, which attacks here on this carbonyl here on carbon 2, and ultimately ends up with this intermediate. So at this time, these electrons here this bond is much more labile than it was before. And the reason for that is that this guy here is in resonance with this bond here. So if it is it's sort of pseudo beta to carbonyl when you have this circumstance, because you have a situation where resonance can stabilize the breaking of this bond. So the electrons resonate to here, that kicks this bond off, making a double bond here, and that's stabilized by these electrons resonating back to nitrogen, and that's enabling this decarboxylation reaction. Now, in the next step, when we're getting ready to transfer this this acyl unit onto a CoA moiety, what happens is sort of the reverse. These electrons from the nitrogen resonate back onto the ring, and it's kicking off the double bond off this acyl unit. Ultimately, those electrons are then transferred onto this lipolamide, which is placed there in the enzyme complex, and that ultimately opens up this ring, or reduces this ring. Now, a base abstracts a hydroxyl off the edge of this, off of this hanging oxygen here, forms a double bond, again allowing the stabilization, or the resonance here allows this reaction to proceed. This guy leaves the active site, and then you end up with your acyl unit attached to your lipolamide. Then a nucleophilic attack from your CoA moiety then allows you to attack this carbonyl, which ultimately leaves you with this dihydrolipolamide, which is actually attached into the enzyme, lysine, and acyl CoA leaving. So now we've made our final carbon product, but we haven't regenerated our reactants. That's what the final step is. So this reduced lipolamide it needs to be oxidized in order to get it active again. So base obstructs this proton. The electrons from here actually go and form a bond here on the sulfur, and that kicks off these electrons, this hydride, which are then attached to an FAD. FAD in chemistry is a cofactor that's like NAD in that it's always involved in redox chemistry. Any reactions involving FAD involve this redox mechanism here. And so this hydride then attacks the nitrogen, that resonates to here, and then ultimately forming our fully reduced FADH2. Okay. So you have a hydride and a proton being picked up, and that's ultimately making our FADH2 and our fully reoxidized lipolamide. Okay, that's ready for chemistry. Now the FADH2 isn't done yet either. It transfers its electrons back onto NAD, and this happens by a base abstracting this proton, resonating to here, which ultimately kicks this hydride off, which is then added on to NAD, similar to the reaction we saw in the lactate dehydrogenase mechanism. And then that resonates down to here and stabilized with the electrons sitting here on a lone pair on this nitrogen. That ultimately makes our final NADH. So the reactions in the TCA cycle proceed through this series of steps that you've learned about previously in your 393 class, but we'll cover in more detail here. As a brief overview, an oxaloacetate molecule from the TCA cycle combines with an acetyl-CoA molecule ultimately to generate this citrate. That thing undergoes this series of transformation, which regenerates oxaloacetate and releases 2 CO2s in the process. What's important to keep track of is every 2 carbons that come in results in 2 carbons going out, along with NADH, ATP, and FADH2. 2 NADH or 3 NADHs, and FADH2 and ATP, or GTP, depending on which nucleotide the enzyme uses. We'll talk a little bit more about that in a minute. So the first step of this reaction is you have acetyl-CoA that's being added on to the oxaloacetate to make the citrate in the citrate synthase reaction. This is a very energetically favorable step, and it occurs through this series of reactions. A base subtracts a proton, ultimately, that makes this enol form of the acetyl-CoA molecule. Then the lone pair of electrons resonate down here, making an excellent nucleophile, which attacks this carbonyl here at this position on the oxaloacetate, the alpha position here, and that ultimately makes this intermediate here. The water then can undergo an additional elimination reaction here, which kicks off the CoA group, ultimately making your citrate bond, or the citrate molecule. So you have a carbon-carbon bond that's formed through this reaction. In the next state, citrate is then converted into isocitrate. And the way that reaction works is that we have the loss and then the gain of a water molecule to ultimately move the position of this hydroxyl from the middle of the molecule off to this carbon 2 position. The way this works is a base subtracts a proton, ultimately going through this enol intermediate. Then a water can then add at the 2 position, ultimately forming this isocitrate. So annoyingly, this molecule is completely symmetric here. It doesn't have a stereocenter, but the isotope labeling data has shown that this isotope, when it goes into this molecule, only ends up in 1 position of alpha-ketoglutarate. Now, this is really important for isotope labeling questions that happen later. If you add an acetyl-CoA group on, it will always end up on the position that's farthest away from the carboxylic acid and the keto group. In other words, it's always adding on the far end of the molecule. It never ends up in the carbon 1 and 2 positions. Okay, even though if you stare at the structure, you should say, well, surely, this hydroxyl should add to either position evenly. It doesn't, and the reason for that is the molecule is what we call is prochiral, which is that the enzyme holds in a particular way. that prevents the chemistry from happening evenly on either side. So I'll actually post a paper about that in the discussion section or on D2L if you want to learn more about it. The important part for isotope tracing is that the carbon is always added to the quote unquote new end of the molecule, which we're going to describe as being the position that's farthest away from the carboxylic acid and the ketone. Okay. So in the next step, isocitrate dehydrogenase is one of the first payout steps here in the TCA cycle. The way this reaction works is a base subtracts a proton off of this carbon 2 hydroxyl in the isocitrate, and that forms a double bond, and that kicks off this hydride that ultimately goes on to NADH. Okay, and that's our first payout step. Then this CO2 then leaves, and that the way this works is this bond right here is this bond right here is beta decarbonyl, so alpha beta. So this guy is labile. These electrons can resonate onto here, breaking this bond, which is stabilized through this enol form. A base subtracts the proton, ultimately tautomerizing this back into the keto form of alpha ketoglutarate. So the pyruvate dehydrogenase reaction has a problem in that it has to break this bond right here, a bond between a carbonyl and a carboxylic acid, which is alpha decarbonyl. So as you know, that's not a energetically favorable reaction, and it requires a cofactor, thymine pyrophosphate. And as it turns out, it uses the exact same mechanism and a series of enzymes as the pyruvate dehydrogenase complex that we learned about briefly before. So that reaction, mechanism you can actually review in the pyruvate dehydrogenase slides above, and it's all the same thing, except we have alpha ketoglutarate coming in and succinyl going out instead of pyruvate and acetyl-CoA, as we did in the other mechanism. Okay, moving along, we have succinyl-CoA synthetase, and what this does is it takes succinyl-CoA, an inorganic phosphate, and a nucleotide, either guanine diphosphate or adenosine diphosphate. We'll talk about there's some difference in isomers that affects which particular nucleotides involved. And then there's a substrate level phosphorylation event that occurs that ultimately makes a GTP or an ATP, frees the CoA molecule and releases the succinate. The way this works is inorganic phosphate acts as a nucleophile, ultimately releasing this CoA molecule in the first step. Then a histidine residue will pick up the phosphate from the carbon chain, releasing succinate. And this leaves this phosphohistidine residue. Then the nucleotide, this can either be guanine or adenosine diphosphate. Then the phosphate on that then acts as a nucleophile, and an additional elimination reaction occurs in which the histidine is regenerated and you get a nucleotide triphosphate leaving. So which nucleotide specifically is generated in the reaction actually depends on the isoform of the enzyme. And there is some substrate level specificity depending on which enzyme's involved. In the context of learning this, is sort of irrelevant, because if a guanine is produced, a guanine triphosphate is produced, there's actually transkinases that'll take this guanine triphosphate and an ADP and make an ATP and a GDP out of it. So there's an easy way to interconvert the nucleotides. So the next step in this in the TCA cycle is succinate dehydrogenase. In this case, we lose a hydride and a proton symmetrically across this molecule, making a fumarate here with this enol in between. This is really characteristic of a reaction catalyzed with this cofactor FAD. Every time you see this chemistry occurring, an FAD is involved. The way this works is a base abstracts a proton, ultimately forming a double bond here, which is too many for this carbon, so it kicks off a hydride. That hydride is actually dumped onto a nitrogen in the middle of this ring here for FAD, And that's possible because these electrons can resonate down, ultimately picking up a proton and making this reduced form of FAD, which has an extra hydride and an extra proton than was present in the oxidized form of the molecule. And then ultimately, we also have this molecule fumarate with the double bond formed. Fumarate can then undergo a transformation into malate. And the way this occurs is that water is added here to the double bond ultimately, and then you end up with this malate molecule here. In the final step, malate is then oxidized and back into oxaloacetate to regenerate our original starting molecule. This works by base subtracting a proton off the middle hydroxyl, forming a double bond, kicking out this hydride, which is added onto NAD. This is possible here because these electrons resonate down, leaving the lone pair on the nitrogen and reforming our NADH, just the same as we saw in our lactate dehydrogenase reaction, and leaving a oxaloacetate molecule here. Now, in the final reflection on this pathway, we can, because of this chemistry happens via a very predictable series of reactions, we can do interesting things with this in terms of biochemistry. If we add fully labeled glucose, which is to say a glucose where every single carbon molecule has an additional neutron on it, so each of these are C13s instead of C12, we can actually track down using a mass spectrometer and watch the appearance of these extra neutrons into these other molecules. So if we have a fully labeled glucose, that'll make a fully labeled pyruvate. That fully labeled pyruvate will make a fully labeled acetyl-CoA, and that, when it's added to an unlabeled oxaloacetate, will make this carbon or this molecule here in citrate that has two additional neutrons on it. So it has a citrate with a mass of +2. So it has two additional neutrons on it that would not normally be there. Because the aconitase has specificity, those two carbons always end up in the same position on oxaloacetate. As we mentioned, those are the two positions that are furthest away from the carbonyl and the keto group. Those ultimately become a 2-carbon succinate and then a 2-carbon fumarate. Now, because the fumarate dehydrogenase reaction or the fumarate hydrogenase reaction doesn't have any symmetry, you end up with labeling scrambling there at the fumarate case. So it can end up on either end of the molecule. The fumarate's totally flat, totally symmetrical, and as it ends up, you end up with labeling in both positions. So that when you go into the second turn of the TCA cycle, new carbon can either be added to the directly adjacent to labeled forms or to the previous labeled forms or not, depending on which of these two oxaloacetate molecules got picked. So as of the second turn of the cycle, you end up with some label scrambling, but in all cases, you will end up with a 4-labeled citrate. Now things get a little more confusing after a CO2 is lost, because on the second turn, then you can end up with 3 labeled alpha-ketoglutarate or 4 labeled alpha-ketoglutarate, depending on which carbon got lost. And then things get progressively more and more muddy as additional turns of the TCA cycle move from there. Okay, so we've talked about the reactions of the TCA cycle and how carbon can be traced through it. What we should touch on briefly is reactions in the TCA cycle that don't come in the form of acetyl-CoA. So there's a series of reactions that are called anaplerosis or anaplerotic reactions, where carbon is added to the TCA cycle, and it results in the net addition of carbon. So remember when we said that when acetyl-CoA comes in, it's balanced by the loss of 2 CO2s. We have an oxidative turn. The same isn't true if you add a glutamine or an alpha-ketoglutarate to the pathway. You end up with the net addition of this 3 additional carbons here, because they're not balanced out by the loss of the CO2 when you fully turn. So this reaction is one example of quite a number of different anaplerotic reactions, reactions that actually contribute net carbon to the TCA cycle. Probably the most famous of these reactions is the Pyruvatecarboxylase reaction. And this is one that's taught most frequently in biochemistry reactions. This is a case where you take pyruvate and a CO2 and ultimately make acetyl-CoA that contributes in net carbon to the TCA cycle. But actually, biologically, probably the two most important contributors are aspartate aminotransferase and alanine aminotransferase, which ultimately allow carbon to come in from glutamate and aspartate, respectively. So we'll learn more about these when we talk about amino acid metabolism, but keep in mind that these are two of the most major contributors to carbon in the TCA cycle, aside from pyruvate. Now, this famous pyruvate carboxylase reaction occurs by taking what we normally is called CO2, but really the reactant that's going into it is bicarbonate, which is the dissolved form of CO2. So bicarbonate doesn't want to be added onto acetyl-CoA by itself. It's an ATP consuming reaction. This reaction is catalyzed by a biotin. Anytime you have a bicarbonate and you're adding it in and you're fixing the carbon into an organic molecule, it uses biotin as a cofactor. The way this works, is that you take your pyruvate moiety here, and this ultimately ends up adding here onto the bicarbonate, and you end up with this 4-carbon molecule here. But importantly, the bicarbonate is added to the position that's farthest away from the carbonyl. So let's talk about some real-world isotope labeling data. This is one of the specialties in my lab, is adding isotopes to cells, tracing around the cycles, and finding out how metabolic pathways work. That's what we do in our group. So I'm going to tell you about an isotope labeling example involving glutamine, where we've added an extra neutron to each of these. So this is 5+ glutamine. And when you analyze this on the mass spectrometer, you see that it has a shift of +5. We call that an isotopomer. Okay, this is what you see in real world. These little blips here from the other channels, this comes from a little bit of contamination because the label that we're using isn't perfectly pure. So when we do an isotope tracing reaction, it looks something like this. We add a glutamine molecule. It ends up with these 5 labeled forms. You end up with this isotope labeling in this position. You can see that enter the TCA cycle here in alpha-ketoglutarate. This way we can conclude that this 5 labeled alpha-ketoglutarate must have originated from this 5 labeled glutamine. Similarly, we can chase this around the pathway and see that ultimately ends up in a 4+ succinate, a 4+ fumarate, a 4+ malate. And weirdly, now when we go further on, we do see a little bit of 4+ citrate, but not a whole lot. And we do see a lot of 4+ aspartate. Now, the reason for this is that malaria cells, where these data were occurring, are behaving in a way that a lot of different human cells behave in. So fibroblasts do this, many cancer cells do this. So it turns out the use of glutamine as a primary substrate for the TCA cycle is a very normal biochemistry. Now, we learn it primarily as oxidizing glucose and converting that into CO2. But actually, most cells, or many cells, use glutamine as a primary source of carbon, and ultimately secrete malate or succinate as major waste products. And that's the case in malaria and also in many cancer cells, is that they'll eat glutamine and secrete malate. And this follows that same logic that I told you about before, of you need to balance the number of carbons in and out. Where we have 5 carbons coming in the form of alpha-ketoglutarate, you have 4 carbons coming out in the form of malate if that's secreted, but you also lose a CO2 in between, right? So ultimately, 5 carbons in, 5 carbons out. Now, what's going on here, if we look at these percent labelings, we see that only a tiny fraction of this carbon actually ends up in oxidative turning in the malaria. And if we look at isotope labeling from glucose, we see that the little turning that does occur here ends up in 2 forms. We see 2+ labeled citrate, but we also say 3+ labeled. So let's talk about this here in the case of oxaloacetate, which is in free equilibrium with oxaloacetate and aspartate are in free equilibrium in the cell. So we actually measure aspartate because it's easier to measure than oxaloacetate. We see both the 2+ label, which makes some sense, and then we see the 3+ label. Now the 2+ label occurs from the citrate synthase reaction where we're taking acetyl-CoA and we get labeled. The 3+ reaction that we're seeing here in malate is occurring from these anaplerotic reactions that we talked about in the Pyruvatecarboxylase reaction. So what we can conclude from this here is, as I showed you before, of the carbon that enters the TCA cycle in malaria parasites, only a small fraction enters from glucose and the majority of it enters from glutamine. And we conclude that, I can conclude that from these data. So what have we learned today? We talked about how carbon enters the TCA cycle, the bridge reactions. We've talked about the specific reaction mechanisms used in the TCA cycle, and we talked about anaplerosis. We also talked about how carbon is not always added in the form of glucose, and I showed you a specific isotope labeling example where carbon is added in the form of glutamine to the TCA cycle.